welcome back. Now, before we proceed, uh, I'm gonna share a picture. So, fear is the path to the dark side. So, this is from Yoda. So, let's now face our fears. Now, for this video, we will look at humoral immunity. So this picture shows us the essence of humoral immunity. Objectives. I think we have discussed the development of T cells. So for this one, we'll focus more on the humoral immune response. So we will look at the basis of antibody production, the different types of the antibodies, and the genetics behind the antibody diversity. Okay, so let's now proceed to humoral immunity. So this humoral immunity is exemplified by when you have an antigen that is presented or is recognized by an immunocompetent B cell, so it will undergo clonal selection. So it will become, it can become a plasma cell. So the plasma cells are the ones that will produce the antibodies. Just remember, for medicine, when we say humor, no, it's not humor sort of funny, but humor in terms of, it means secretions. So in this case, there is a secreted antibodies. So this one you've shown, you've shown this. So this one, I've already shown this in the previous video, I think, in the development of B cells. So the immunocompetent B cell, after its sort of education from the bone marrow or in the bone marrow, it will now travel towards the secondary lymphoid organs where it will reside. Okay. Here. So this is the lymph node. Now in the lymph node, it will undergo clonal expansion and diversification of its receptors. Okay. Now, in the presence of an activated T cell or in the presence of an antigen, okay, this B cell or this activated T cell can sort of wake up this naive B cell, or it can sort of activate this B cell. Now, upon activation, it will undergo differentiation and clonal selection. So it can either become a plasma cell, the one responsible for the secretion of the antibodies, or a memory B cell. So as I mentioned before, there are two ways by which the B cells can be activated, either by directly interacting with the antigen or by the use of an activated helper T cell. So there's actually a difference. If it is a proteinaceous antigen, it is more of through the activation of helper T cell. If it's another type of antigen, example would be carbohydrates and lipids, the B cells can directly interact with it. Okay. The B cells can recognize those types of antigen. Now the difference here is that usually those that are because of helper T cell activation tend to be more long-lasting or longer-lasting compared to this type of B cell activation. Now, once activated, the B cells can differentiate into a plasma cell, the one that secretes the antibodies, and memory cells. Take note, the memory cells here is somewhat similar to the memory T cells. So they do not react right away, but they act as a reserve, such that when we encounter the same microbe or antigen later on, these memory cells can rapidly act. So memory. So they do not forget, unlike Dory. Okay, so this is one way to activate the B cell. So let's look at the steps one by one. So first one is, this, same as with the cell-mediated immunity, the antigen is being presented by the antigen-presenting cell. So it will be displayed at the surface together with the class 2 MHC. And then this will be recognized by the CD4 T cell or the helper T cell. Now, one of the things that this helper T cell can do is it can activate the B cells through the release of interleukin 2. And then once activated, the B cell will differentiate into plasma cells and memory B cells. So these are the phases of the humoral immunity. Take note of the outcomes here. So possible outcomes after the activation of the B cell would be, number 1 would be antibody secretion. This is, again, a function of the plasma cells. Another thing that can happen is what we call as your isotype switching, wherein the antibody being secreted will be switched from one type to another. Example, from a type M, IgM, it will become an IgG. Another thing that can happen is affinity maturation, wherein in the long run, what happens is the B cells will develop from a low affinity to a high affinity antibody expressing B cell, such that the antibodies produced at this point would have a stronger binding or stronger interaction compared to the previously secreted antibodies. And then another is, because of this high affinity antibody expressing B cell, it can branch off into a subset of that would be the memory B cells. Okay, before we proceed, please, you can pause this video and you can copy this table. Okay, so give examples or conditions that would best represent the different types of acquired immunity. So let's look at it one by one. So first one, when we say active humoral immunity, it means to say that the B cells would encounter the antigen, or the adaptive immunity directly encounters the antigen, and then the B cells will be the one producing the antibodies against this antigen. So it means to say that our body is actively producing the antibody against the antigen. As compared to passive humoral immunity, wherein there is an introduction of a non-native antibody. Okay. Non-native meaning to say an antibody coming from another source or another person is given to the person. Another way to classify immunity is if it is naturally or artificially acquired. When we say naturally acquired, it means to say that the antigen or the antibody is given to that person in his daily life, okay. Naturally, in other words. So it can either be a natural exposure to an antigen or a transmission of the antibodies from mother to the fetus during pregnancy. Artificially acquired immunity is through the introduction of vaccines. Now just take note that our vaccines can either be antigenic or the vaccine can be an antibody itself. Okay, so when we combine this classification together, we'll have 4 types. So we have your naturally acquired active immunity. So when we say naturally acquired active immunity, the antigen or the microbe or the pathogen enters the body naturally. Okay. And then the body, and then that person's body will generate the immune response against that antigen. Example would be the childhood illnesses. I'm sure you've had this, the chickenpox or varicella, mumps, measles, German measles. Okay. So they would confer lifelong immunity. So we can only have this illnesses or sickness only once, because we have the memory cells for this. The other type of natural acquired immunity would be the passive immunity, naturally acquired passive immunity. So example of this one is the transmission of the antibodies from the mother to the fetus, okay, during pregnancy, or the passing or the transfer of the antibodies from mother to the baby during breastfeeding. Now, the difference here from that of the active immunity is that since the antibodies are the ones being transferred, the immunity is usually short-lived. At most, probably 6 months. Yeah. It would only confer protection until the child's immune system can start producing the antibodies. The other type would be the artificially acquired immunity. So again, there are 2. You have your artificially acquired active immunity. Again, when we say artificially, it's through the vaccines. Okay, first type would be if the vaccine given is antigenic. So it would confer an active immunity. The other one is your artificially acquired passive immunity, wherein the vaccine given is an antibody, whereas an immune serum. Again, the difference here is that active immunity would have a longer lasting immunity. For passive, could only last for weeks to several months. And for passive immunity, the host immune system does not respond to the antigens because we do not form the memory cells for this one. Okay, so again, reminder to stretch. Okay, you can pause and you can stretch. Okay, before we proceed, I have a question. So why don't ants get sick? Yeah, it's because they have little antibodies. Anyway, let's now proceed to the antibodies. So these are gamma globulins. So they are proteins, they are plasma proteins. So approximately they would comprise 20% of our total proteins in the blood. So other name of the antibodies would be the immunoglobulins. That's why in the books, they are termed as IGs, immunoglobulins. Now they would act by, number 1, by blocking the microbes from binding to and entering the host cells. They could also bind to microbial toxins, thereby neutralizing the effect of these toxins. And last one is they could also directly eliminate the microbes, toxins, and infected cells through several mechanisms. However, take note that the antibodies, they cannot enter the cells. So they are not effective against microbes or pathogens that live inside the cells. However, these antibodies can still perform their function to protect or as a defense, because these antibodies can still bind to microbes before they can enter the host cells. Or during the passage from the infected to uninfected cells, or rather, during the passage of the microbe into the uninfected cell, these antibodies can still bind to these microbes. Okay, so this is the antibody structure. Okay, so it's Y-shaped, and you have two sets or two pairs of chains. You have the longer one, which is called the heavy chain, and the shorter one, which is called the light chain. Aside from that, we also have the variable regions and the constant region. Now, what's the difference here? The variable regions would vary depending on the antigen. It is specific, or it would vary depending on the antigen that this antibody is specific. For the constant chain, so it's the same for all types of antibodies. Another part would be the Fab region, or what we call as your antigen binding fragment, because initially they discovered this as a that the antibodies would have two fragments. So they would cleave this part, the black part, which is hydrolyzed by certain enzymes. So they would have the Fab region here, and then the other region. Now, this Fab region is the antigen binding fragments, or this is the one that binds to the antigen directly. Okay. Take note, parts of the Fab region is that you have the variable components of the light chain and heavy chain, as well as a portion of the constant light chain and constant heavy chain. So this is the one that binds to an antigen. This is another Fab region that can still bind to an antigen. Another part would be the crystalline fragment. Okay, so this crystalline fragment is constant again for every antibody type. So it's composed of these heavy chains, constant part. Now, this Fc region is the one that is being recognized by other immune cells. So it's sort of like it's a tag. Okay. So if this one binds to the antigen, this is the tag. The other immune cells can immediately see now that that microbe is being tagged by your antibody. So this is for recognition of the antibodies. Now, as I mentioned earlier, being the antibodies being proteins, okay, they would also undergo the process of protein synthesis. So, transcription from DNA to mRNA, and then translation from mRNA to the peptide chain. Okay. So this is in the rough endoplasmic reticulum, and then after that it will undergo post-translational modification to become the active antibody. This would give us an idea of why we have the different sets of antibodies, or different types of antibodies. Now, the one that would dictate which type will be predominate predominantly produced would be the cytokines. Example, for interleukins 4 and 5, if it is simultaneously secreted, first one is it will produce IgM. You will know later on why IgM first. And then, after some time, it will be the IgG. If it's IL-5, you have the IgA. If it's IL-4, it's IgE. Now, the different types of the antibodies would be the mnemonic is GAMED, GAMED. Okay. We will look at these different types of antibodies later on. For this one, we have different mechanisms how these antibodies work. So we have your neutralization and agglutination. So this is neutralization, okay, wherein the microbe is neutralized, or the toxin is neutralized. And we also have agglutination, wherein the bacterial cell or the pathogen are clumped together. We also have opsonization. I think you already know this opsonization from the previous videos. Okay. Yeah. So it makes these antibodies would coat the microbial cell, and this would enhance the process of phagocytosis. And then we also have the activation of complement. Remember the classical pathway. The trigger for that is the antibody binding to the membrane of the bacterial cell. And then we also have cell lysis, wherein because of the antibody binding to the membrane of the microbe, this will be tagged, and this will be recognized by natural killer cell or cytotoxic T cells, so thereby killing that bacterial cell. And it could also induce inflammation. So these are the immunoglobulin types or antibody types. So first one is your IgA. So take note that the IgA, it can exist as a monomeric, dimeric, or trimeric form. When we say monomeric, it's the Y-shaped. Okay. So this picture is a dimeric, so you have two Y shapes here, two monomers. Okay. Now, what is important about IgA is that it is the one that confers mucosal immunity. So it is the one secreted together with the secretions. Okay. And it can pass through the breast milk. So this is the dimeric form. Just take note that it has this pentagon here. This is the J junction or the J protein. Now, for it to pass through the mucosa, it must be in the dimeric form. The other type would be IgD. This is easy. The function is still unknown, but it is expressed together with IgM. It is expressed on the surface of B cells. So in other words, this can act as a B cell, or this is a part of the B cell receptors. The other type is the IgE. Okay. So it exists in the monomeric form, Y shape only here, and its function is for mast cell activation. So the mast cells here, this is an example of a mast cell. Located in this mast cell is that it has Fc receptors. So when the IgE binds to a pathogen, and then the IgE, the Fc portion of that is recognized by the mast cell will degranulate or will release its granules containing histamine, thereby initiating the hypersensitivity reaction. So there will be vasodilation and increase in vascular permeability. The way I remember this is letter E stands for allergic response. Next one would be the IgG. So for the IgG, it is still monomeric, okay? And these are the functions of the IgG. There are actually a lot because this IgG is the most numerous in terms of the number of your antibodies in our body. Now, one function of the IgG is it can act as an opsonine, so it can cause opsonization. It can coat the bacterial pathogens or the pathogens and prevent them from entering host cells. So this is an example of coating of the IgG. So the pathogen cannot enter the cell. Okay, aside from that, the Fc portion of this IgG that is bound to an antigen can be recognized by a macrophage, thereby phagocytosing the antigen. So this is the process of phagocytosis. Another thing that can happen is what we call as your antibody-dependent cellular cytotoxicity, wherein the antibodies again bind to the surface of the bacterial cell, and then these antibodies would be detected by the natural killer cells. Yeah. So thereby inducing apoptosis for this target cell. Another thing is that another important feature or characteristic of the IgG is it is the one that can cross the placenta. So this is the maternal blood, this is the placenta, and this is the fetal blood, or the blood of the fetus. Okay, so the IgG can be endocytosed by the placenta, and then once inside this endosome, okay, it will bind now to an Fc receptor. So the Fc portion will bind to it. Now this binding of the IgG to the Fc receptor will cause the exocytosis, or will signal the exocytosis of the IgG into the fetal blood. So it sort of travels from the mother to the fetal blood. Okay, last subtype would be the IgM. So take note, the IgM, it exists as in a pentameric structure. Pentameric. And these are the functions of the IgM. Okay. So it's pentameric, meaning to say, so it has 5 monomeric units, and in each monomeric unit you have two antigen binding sites. So for IgM, you have 10 different antigen binding sites. So it can bind to a lot of antigens. And same way as with IgA, it has this pentagon at the center, which is the J chain or the J protein, okay, which is also for secretion. And take note of this, IgM is the first antibody expressed in a mature immunocompetent B cell. Okay, so it is the one that is being produced when a B cell binds to an antigen, or when a B cell is activated. Aside from that, IgM is also expressed on the surface of the B cell. So it is also a part of the B cell receptor. So my mnemonic for IgM is that M stands for malaki, big, because it is pentameric in structure. Aside from that, it's mabilis, because it is the first one that is being produced after exposure to an antigen. And mauuna, mabilis or mauuna, and mabilis din mawawala. So its half-life is very short. So malaki, mabilis, mauuna. So again, summary of the antibody types. Okay, now let's look at the diversity of the antibodies. So, we only have 5 types of antibodies, but these antibodies can react to a very specific antigen. And I think you already know by now that there are a lot of antigens. So, meaning to say, we should have a diversity, a diverse set of antibodies. So, first process, so there are actually two processes. First one is what we call as your somatic recombination. So we're in the, meaning to say, these antibodies, the genetic material for this one is not passed from the mother, it's not passed on to the daughter cells. So it's not inherited, in other words. So this would give us a sort of concept of why immunity is not passed from mother to child, but rather immunity is developed throughout the lifetime. Okay, so again, remember the antibody structure, okay, light chain and heavy chain. Okay, so in our DNA, okay, in the DNA, so there are areas there that would code for specific proteins. Okay, so we have here the area that would code for the light chain, and then the area that would code for the heavy chain here. So just remember the colors. Okay, light chain, heavy chain. Now, on this heavy chain, on this part, okay, actually, this part, the variable region of the heavy chain. On the variable region of the heavy chain, you have 3 different areas encoded in the DNA. So there are 3 areas. This one, the V, would stand for the variable region. D stands for the diversity region, and J stands for the joining region. Now, all of this would have different combinations. Okay, for example, this one. So, for example, you have 40 sets of the variable region, 23 sets of the diversity region, and 6 sets of the joining region. Now, when we combine this all together after several processes, you can produce approximately 5,500 combinations of this area. So imagine, just by changing these 3 regions, the variable region, diversity region, and joining region, just by changing this, we can have approximately 5,500 sets of variable regions. Okay, for the heavy chain. But remember, we also have the light chain. So for the light chain, there are actually 350 combinations for this one, for the variable region of the light chain. Such that when we combine this together, it's a lot. 350 times 5,500, that would be for approximately, just do the math, okay? That's just one process. So this is your somatic recombination. Another process is your somatic hypermutation. Now, located again on the variable regions, what happens here is that there could be mutations on the coding region. So here. So again, in your variable region, a joining region, and diversity region of both the light chain and heavy chain, okay, there could be minute mutations. So when there's a mutation, the protein expressed there would be different as well. Okay. So resulting now to a different type of antigen-specific antibody. So again, two processes for the diversity, somatic recombination and somatic hypermutation. Okay, so in summary, what we have discussed so far would be the number one, features of the humoral immunity. Okay. We've also looked at the different types of immunity. Now, just remember, if it's natural, we get it naturally. Artificial, we get it through vaccinations or introduction of non-native antibodies. Active rather, is our body makes the antibodies against that antigen. For passive, an antibody not from our own body is introduced. Okay. We have also looked at the different sets of antibodies gamed, GAMED, and their respective functions and mechanisms. And then we also looked at the genetic basis of the antibody diversity, your somatic recombination and somatic hypermutation.